Hello students, welcome to EPG Pathshala. I am Vidya from All India Institute of Medical Sciences. I also worked at JP Institute of Information Technology, Noida, and was director of Dr. D.Y. Patil Biotechnology and Bioinformatics Institute, Pune. My main identity is I am a teacher. Today, we are going to discuss about a module, DNA supercoiling and hyperchromicity, under the paper Biomolecules and their interactions. What is supercoiling? We all experience it somewhere or the other. In elastic bands, ropes, electric wires. This is a very common phenomena. And how do we remove it? There are various ways. Either we start from one end and start unwinding, or we cut it, and so on. Supercoiling of DNA is defined as over or undercoiling to DNA to remove strain. It is a helix upon helix structure, or called the tertiary structure of DNA. Supercoiling is extremely important for packaging of DNA molecule, which is several meters in length, and to fit it into the chromatin, which is 200 to 300 angstrom wide and 700 to 800 nanometers in length. I want to also describe various physico-chemical techniques that are used to study DNA supercoiling. So let me explain you what you are going to learn in this module. I want to introduce first to the existence of bending and coiling and supercoiling in DNA. Next, I want to brief you on the mathematical expression for DNA supercoiling. Next, we will discuss DNA supercoiling in nucleosome core particle. Later, I want to explain by what is hyper and what is hypochromicity in DNA. I wish also to introduce you to CD spectroscopy, which is extensively used to study polymorphic structural changes in DNA. DNA bending and supercoiling. As seen earlier, DNA molecule has an intrinsic ability to bend as a direct consequence of change in the base composition. This was shown in the X-ray structure of a DNA sequence CG, CG, AA, TT, CG, CG by Dickerson's group, where the DNA molecule showed bending of 19 degrees. Existence of supercoiling in nature is always seen in case of circular DNA. Circular DNA is generally present only in a supercoiled state. We show here a relaxed double helical segment of DNA. There are various different supercoiling in circular DNA. If two ends of DNA are closed into a circular DNA and then allowed to retort freely, DNA goes into a figure of 8 and many such figures. And the superhelical structure with few low rethes, we will explain it later, is shown in this picture. Like first, there is no supercoiling, then twist -1 rethe 0, then twist 0 but with -1, like twist +1 with 0, twist 0 with -1, and some higher order, twist 0 with -4, or twist 0 with +2, twist 0 with -2, and so on. Very interesting structures are formed. Mathematical expression for circular DNA. Circular DNA is always isolated as a superhelix with tertiary winding. Right-handed is a relaxed or open circular structure. For the purpose of computation, right-handed superhelix is defined as having a negative number of superhelical turns, whereas left-handed superhelix is defined as having a positive number of superhelical turns. We show in this picture a negative supertwist. Both secondary and tertiary windings shown in this figure are right-handed, and the supertwists are negative, equal to -3. Mathematical expression for defining superhelical structure. DNA supercoiling can be defined as a change in linking number Lk, which is the most descriptive property of the superhelices. We define a linking number Lk0 as the number of turns in relaxed B-form DNA plasmid molecule. This number is obtained by dividing total number of base pairs BP in the molecule by relaxed base pairs per turn, which is normally taken as 10.4 to 10.5, and the ratio is called Lk0. So thus Lk0 is base pair number divided by 10.4. Now Lk is the number of crosses a single strand makes over other. It is known as the Watson-Crick twist found in the circular chromosomes when it is constrained to a plane. Lk is given equal to Tw plus Wr, where Tw is the number of twists, Watson-Crick twist, when the chromosome is not constrained to a plane, and Wr is the number of coils or rethes, is the number of superhelical twists. This is how rethe or Wr is defined. We like to give energy expression, the Gibbs free energy. Gibbs free energy delta Lk, which is the difference between Lk and Lk0, that is Lk minus Lk0. But then we define a property which does not depend on the number of base pairs. So we call it sigma, and sigma is equal to delta Lk divided by Lk0. Then delta G upon N is taken equal to 10 RT into sigma square. The difference between the supercoiled circular DNA and non-coiled circular DNA with N greater than 2000 base pairs is given by delta G by N, which is 700 kcal per divided by base pairs multiplied by delta Lk upon N, which is generally around 16 cal per base pair. A very classic example of DNA supercoiling exists in nucleosome. What is nucleosome? Nucleosome is a basic unit of DNA in eukaryotes, consisting of a DNA segment which is about 200 base pairs wound around an 8 histone core protein. Nucleosomes are fundamental repeating units of chromatin, which packs about 2 meters of linear DNA in 10 micrometer nucleus. These are folded into series of higher order structures to eventually give you chromosome. Nucleosome structure. Nucleosomes were first observed by Olins and Olins in 1974 in electron microscopy photographs. Three-dimensional structure was proposed by Kornberg in 1974. Alan Klug provided for the first time the evidence that 140 units of DNA molecule wrapped around 8 histone protein core as a left-handed superhelix in 1/3-port turn with external diameter of 110 angstrom. Actually, this was done using image reconstruction coupled with electron microscopy technique, and one of the most interesting structure present at that time. We show this picture of nucleosome. Here there are 4 histone pairs, H2A, H2B, H3 and H4. So total 8 histone core proteins shown by blue balls. This figure is taken from Strier's book of biochemistry. The DNA molecule, 140 base pairs, shown in red, wraps around this 8 histone protein core. Then histone H1 is attached at the end. There is a linker DNA shown in dark red color, which is covering the histone H1, the linker histone. We show a beautiful picture of nucleosome in this figure, because from 1974 to 2000, because of the progress in crystallographic and analysis and several other techniques, the detailed 3D structure at atomic resolution of nucleosome has become available. We show in this picture a PDB structure 1EQZ by Harp, which was discovered in the year 2000, and the picture is drawn by Python molecular viewer. Here we see the DNA molecule shown in the black color and the histone shown as a cartoons by a variety of colors. In the other also we show even the linker DNA and histone proteins in the beautiful top view of the histone structure. Protein DNA interactions. The histone protein has over 120 protein DNA interactions with DNA which is wrapped around it. And these interactions are unevenly spread in the regions alpha 1, alpha 2 and L1, L2. The alpha 1, alpha 2 site uses 2 adjacent helices, whereas L1, L2 site uses 2 adjacent loops. Salt links and hydrogen bonding between protein side chains and basic and hydroxy groups and main chain amides with DNA backbone phosphates form the bulk of interactions with DNA. Histone DNA interaction. Some interactions are specific. However, most of the interaction amongst histone protein and DNA are non-specific. Latter is very important given ubiquitous distribution of nucleosome along genomes. Although nucleosome prefer some DNA sequences over other, these are practically non-specific and this can bind to any DNA sequence. Non-polar interactions. Non-polar interactions are made between protein side chains and deoxyribose groups. Arginine side chain intercalates into the DNA minor groove at all 14 sites where it faces the octamer surface. The distribution and strength of DNA binding sites about the octamer surface distorts the DNA within the nucleosome core. Distortion in the DNA. The DNA is non-uniformly bent and also contains twist defects. The twist of free form of DNA in solution is 10.5 base pairs per turn. However, the overall twist of nucleosomal DNA is only 10.2 base pairs per turn, but this varies from 9.4 to 10.9 base pairs per turn. The histone tail is flexible. The N-terminal tail of histone H3 and H2B pass through a channel formed by minor grooves of two DNA strands. How does histone H4 interact? The N-terminal tail of histone H4 has highly basic amino acids. This interacts with H2A, H2B dimer of outer nucleosomes and is important for higher order structures. Linear histones such as H1 and its isomers are involved in chromatin compaction and sit at the base of nucleosome near DNA entry and exit binding to the linker region of DNA. The structure of core particle with linker looks like beaded strings, very beautiful. This is shown in the picture here. These histone proteins are shown in purple color. The DNA which is wound on it is shown in the black color, and how the whole thing ultimately fits into the chromosome is shown schematically. Sedimentation coefficient measurement is a technique for measuring supercoiling of DNA. Supercoiling can be ascertained by a number of techniques such as spectroscopy, low-angle X-ray diffraction, electron microscopy, and measurement of sedimentation coefficient. A typical example of sedimentation coefficient measurement will be seen in the next couple of slides. Sedimentation coefficient measurement, it can be used for finding the DNA coiling. We show in this picture the form 1, which is shown in a blue curve, is a traditional native form of duplex circular DNA, and the sedimentation coefficient SW is plotted on the y-axis for various pH and the x-axis. If one or more nicks are involved into the DNA, then there is a free rotation of one strand with respect to other, and it forms form 2, which shows the sedimentation coefficient variation given in the red color. The form 4 which is shown in the green curve, is the product of alkali denaturization of form 1. It is only the form 1 which shows a dip at a particular pH value. Between pH 7 and pH 11.5, the sedimentation coefficient for form 1 is constant. Then it dips, and at just below 12, it reaches a minimum value. But with further increase in the pH, it returns back to its original value, but it doesn't stop there. It continues to increase relentlessly. And by pH 13, the value of S has risen to nearly 50, 2 or 3 times the value at pH 7, indicating an extremely compact structure. DNA hyperchromicity. Ultraviolet UV spectroscopy is one of the most important tools in understanding structural changes and interactions of DNA. Generally, we measure OD, the optical density, as equal to log to the base 10 I0 upon I, or we measure transmittance, which is I upon I0 multiplied by 100. These are usually governed by Beer-Lambert's law, I equal to I0 exponential to the power minus epsilon Cl, where epsilon is molar absorptivity and C is the concentration, while L is the path length. The spectral lines show changes due to various interactions. First, let us see the nomenclature for these changes. Pathochromicity is a shift towards higher wavelength or redshift. Hypsochromicity is the shift towards lower wavelength or blue shift. Hyperchromicity is increase in absorbance. Hypochromicity is reduction in absorbance. We show in this picture UV absorption of DNA bases adenine, guanine, cytosine, and thymine. You can immediately notice the UV absorption curve of these 4 bases, that there is a crowding of peaks around 260 nanometers. UV absorption and single and double stranded DNA. We show here 2 curves, red for double stranded DNA and blue for single stranded DNA, giving the UV absorption. The double helical B form of DNA has a strong ultraviolet absorption at 260 nanometers due to n-pi star transition. The average extinction coefficient for double-stranded DNA is 0.002. But for single-stranded DNA, it increases because of the hyperchromic effect. It becomes 0.025. Hyperchromic effect, DNA molecule shows hyperchromic effect when the solution of DNA is heated above its melting temperature, 80 degrees. The double-stranded DNA unwinds to form single-stranded DNA and absorb more light. And hyperchromicity can be used for quantitizing DNA melting. To quantize DNA melting, we define some parameters for hyperchromic effect in DNA. We define hyperchromicity A as equal to 100 multiplied by ET minus EC divided by EC, where ET is absorptions at temperature T, and EC is absorbance at 100% coil, which is used to monitor helix coil transition in DNA. We show in this picture how the absorption changes in different DNA structure sizes and winds. Like on the left hand side of this figure, we show a double-stranded DNA and single-stranded DNA, and on the right hand side of the picture, we show two curves, one a continuous curve, which is a lower curve, which is for double-stranded DNA, and or a dotted curves for denatured DNA, and the DNA is showing here the hyperchromic effect. Hyperchromicity and DNA melting. The transition melting temperature Tm is defined as the temperature at which UV absorbance of the light is 50% between the maximum and minimum, where 50% DNA is really denatured. That can be seen in this picture here, where we show the value for single-stranded, which is 100%, and double-stranded, and the midpoint of the two is taken as the transition temperature. Hyperchromicity can be used to track condition of DNA as temperature changes, and relative absorbance is absorbance at any temperature T divided by absorbance at 25 degrees. There is striking increase in absorbance upon denaturization of DNA. Monitoring polymorphic structural changes in DNA using CD spectroscopy. Circular dichroism is a phenomena originating from interaction of chiral molecules like DNA bases with circularly polarized electromagnetic rays. The absorption of left and right-handed circularly polarized light by chiral molecules differs, and the difference can be measured as molar ellipticity theta. But generally, we measure the difference in molecular extinction coefficient for left and right circularly polarized light, delta epsilon equal to epsilon L minus epsilon R. Now, this parameter is extremely important because it depends on the conformation of DNA. We show in this picture 3 CD spectra for B form of DNA, C form of DNA, and A form of DNA. In the case of B form, you see first crossover at 260 nanometers, but at lower value, there is a negative peak, and at higher value, there is a positive peak. In A form, it is similar to B form, but the negative peak is very, very small, and the positive peak is very, very large. In B form, they are more or less of equal magnitude. In the C form, the negative peak is far bigger or far lower, we can say, and the positive peak is practically absent. So by looking at the CD spectra of the DNA, you can immediately tell whether it's a B form or a C form or A form. Between the G form, A form and B form, also there is a drastic difference rather. Actually, the G form of DNA was first discovered in the CD spectroscopic studies by Paul and Gibb. And he got two spectra, which are shown on the left-hand side. bDNA, which is shown in a continuous slice, and zDNA in a dotted. And the spectra was just inverted. And based on that, he proposed that DNA may be having a different handedness than B form of DNA. And later on, it was proved that that is a zDNA. In the right-hand figure, we show three figures, lines, curves. Red for B form of DNA, blue for A form of DNA, and green for Z form of DNA. And we find the inversion and differences which are used customarily for understanding polymorphic structural transition or BZ transition, AB transition in DNA. So students, what we have learnt in today's presentation? We have learnt first what is DNA bending and what is DNA supercoiling. We have seen how to quantitate DNA supercoiling, the mathematical expression used for DNA supercoiling. Then we have seen a practical example of DNA supercoiling in the case of nucleosome. This is a typical way by which DNA molecule is compact and then it can fit into the chromosome. We also saw in today's lecture use of sedimentation coefficient method for studying DNA supercoiling. I introduce you to UV spectroscopy and various parameters of UV spectroscopy such as hyperchromic effect, hypochromic effect, then how these effects can be used for studying DNA melting, interaction of DNA with other molecules, interaction of DNA with ions, and so on. We have also introduced you to CD spectroscopy for DNA interaction. This is a very powerful technique, and the interaction of various small molecules, carcinogens, drugs with the DNA molecule can be followed using CD spectroscopy technique, and one can very easily see how the polymorphic changes in the DNA occur and what will be their implications under different environmental conditions. Thank you.